Thank you very much, and thank you very much for Duncan and Damien and others at Sofian for inviting me. I'm really sorry I can't join you for the dinner tonight, but I do remember from the last century when I was a student here that Clare College does excellent food and also some of the best music nights that you'll get in Cambridge. So hopefully that's still true today, so you'll enjoy that. So what I want to talk to you about today is a class of ion channels which perhaps not many people are too familiar with, and that's ion channels encoded by viruses, of which there are many. And specifically work we're doing to try and generate proof of principle that these are expeditable drug targets because of the need for antivirals in different circumstances. I'm going to focus on Zika virus here. This is not a brain, this is a Zika virus particle. This is not chorizo, it's a blood cell. But yeah, we have many different examples of which. So we'll start by talking roughly about what these ion channels are, just to introduce the idea, and their feasibility of drug targets. And then, of course, I'll focus on our Zika virus story, which I was due to present last year as sort of unpublished, but is now published at the end of last year. So viroporins, they're small hydrophobic proteins, and they're encoded by a myriad different classes of viruses, and they have multiple different functions, as this cartoon is meant to show you here. But primarily, the ones that we're interested in looking at act during these assembly and virion entry pathways. And they do this by remodeling cellular secretory pathways and also by altering ionic gradients during endosomes, which serves to sort of prime these virus particles for entry. And the most famous of these is the influenza M2 proton channel. But as you can see here, we're classifying our Zika virus M protein, which I'll tell you about in a moment, as also part of this process going in and out of cells. Okay? They're all very small, they all have to oligomerize, and they have between 1 and 3 different transmembrane domains in general. But the problem is that these are very poorly studied, and there are a very, very large number of studies claiming that basically every kind of membrane-disrupted protein that viruses make acts as a viroporin. So they're sort of considered to be this sort of virus encoded ion channel poor dualism. But actually, this sort of biphasic activity is more to do really with how well they've been studied. And it turns out that we understand the gating of the ones that have been well studied, and we don't really understand the gating of the ones that haven't been well studied. So that bias is there. And this sewer pipe reference actually comes from the first viroporin grant submission I put into the MRC, where one of the reviewers described the Hep C ion channel that I was working on at the time as a sewer pipe, which was lovely. But what we tried to do, and this is a publication we published last year with Mette Rosenkild and colleagues in Denmark, of trying to sort of make the field more rational and look at people's different experiments very logistically to try and determine which ones are viroporins and which are not. But as I said, the ones that have been well studied, and this is the archetypal viroporin, the M2 proton channel from influenza virus, we actually have a pretty good molecular understanding of this very simplistic, although very effective, monodirectional gating of protons through this channel through protonation of the histine 37-imidazole, which causes structural shifts, moving this gating trip residue out of the way. Okay? So where there's lots of structural information, where there's lots of experimental work, we actually understand these proteins pretty well. However, the ones we're working on, we don't. So please forgive me. So why are we working on these? Okay, so I think that if the pandemic continues to tell us anything, it's that we really don't have enough effective antivirals for viruses that are going to cause us problems that are surprising. So we have lots of drugs against HIV, we have lots of drugs against Hep C, etc., all very Western problems. But against emerging viruses, persuading a drugs company to make a drug that might get used is a difficult ask. And really, governments should be picking up this bill, but they don't seem to be particularly, and all the government schemes that we're familiar with actually angle themselves towards marketability. So that when it came to 2020, trying to look at drugs that would stop COVID, the shelves were pretty bare. And this drove a sort of fairly desperate response in terms of drug repurposing and led to some very, very strange drug confirmation and approval systems, which I don't think we really need to talk about now. But we believe that these viroporins are actually a really good idea for drug targets and something which can be exploited along the lines of these sort of 100-day plans where we can generate inhibitors of something that's ready to go in a phase 2 trial should the need arise. And we think this because actually the M2 channel that I mentioned before was actually the target of one of the very first antiviral drugs licensed in the 1960s, amantadine, although they didn't actually realize this was the target at the time. Unfortunately, this is a bit of a double-edged sword because these adamantane drugs, amantadine, here I'm showing you, here is just literally a greasy blob with a primary amine on the top. These drugs have incredibly poor drug-like qualities, and this led to very high levels of resistance almost immediately upon using them. So whilst people actually understand this as a clinically viable drug target, unfortunately the type of inhibitors that people used as drugs have given these drug targets a bad name in a lot of pharmacological circles. But we believe that modern drug discovery is a bit better than this. So that's what we're really trying to do in the lab, to try and show that we can rationally develop inhibitors of these virus ion channels. It's difficult because we have limited structural information, plenty for this guy, not really for anything else. No full-length sequences, unfortunately, and their expression is extremely challenging, and really only the flu-M2 protein goes to the cell surface. So unfortunately, using these wonderful patch clamp systems at the moment for the ones that we're working on is quite difficult. But we do know that they're very useful as drug targets, and potentially because of that role in going in and out of the cell, they could be used as antiviral prophylaxis. And these are just a list of the viroporins that we're working on at the moment. We tried to pick the nastiest viruses possible, obviously. I used to work on hepatitis C a long time ago, but there's no money for that now because apparently it's cured. But we're working currently on flu. Zika is an example of flaviviruses, which I'll come on to at the moment, and of course, SARS-CoV-2 and other coronaviruses. So I've just been slagging off rimantadine, and yes, we can call it a bit of a pharmacological dunce, but it's actually really, really useful because of the promiscuity of these drugs. We can use this to identify one of these so-called simplistic channels actually has a druggable channel activity, because if you throw enough rimantadine at something, it's going to stick, okay? So what we actually do is we use rimantadine as a sort of first pass to identify druggable channel activities and use these to then develop more rational early hit stage inhibitors that we can then use both in sort of looking at structure determination, looking at potentially antiviral projects, but mainly, because I'm a virus geek, we want to understand what these channels are doing in the virus life cycle. Now, the one we're going to focus on today is Zika virus. Why Zika virus? Well, Zika virus is an example of flaviviruses, which are arboviruses, i.e. arthropod-borne viruses. And so these are transmitted by mosquitoes, ticks, what have you. And we're interested in Zika because this is an example of a re-emerging virus infection. It was found to be existing in Uganda in the 1940s in the Zika forest, and really was only documented as causing a relatively few very mild infections in humans for about 50 or so years. However, in the early noughties, people started to know that different strains of this virus were spreading eastward across the planet, and all of a sudden we started getting these outbreaks in Yap, in Micronesia, and in other places, which eventually culminated in this massive outbreak, if people remember, about 10 years ago in South America. And this was really bad because this virus is changing pathogenicity as well as causing an increase in transmission. So we really had nothing to counter this apart from vector control, so spraying for mosquitoes. So this is a real problem, and these viruses get into human populations when the mosquitoes decide not to bite their standard hosts, but decide to bite human hosts. And then we have these Aedes species mosquitoes which propagate the spread in cities. And of course, in Brazil, these mosquitoes are hugely prevalent, and they cause a huge amount of disease. And I mentioned the increase in transmissibility. These are the mosquitoes that are spreading these things, these Aedes species mosquitoes, pretty nasty, day-biting mosquitoes, not very pleasant. And what we noticed in Brazil, of course, was this terribly tragic outcome, particularly in mothers that were infected during pregnancy, where this virus goes to infect neural progenitor cells during neural development and causes these really astonishing, horrible outcomes in terms of fetal microcephaly. But generally, Zika virus causes a relatively mild disease, similar to most other flaviviruses, but the neurological complications have both worsened and become more common in these new strains that are spread across the planet. So this is why this is a re-emerging source of infection. And just to make people aware that this is not just a developing world problem, because climate change is increasing the range of these viruses and the mosquitoes specifically that transport them. And particularly these Aedes aegypti mosquitoes are spreading out from the equatorial regions, and it's predicted that at least by 2050 it's going to be in Northern Europe. And if anything's going to make the UK government fund projects on this, it's certain that the south of England will be affected before the north of England. So that's fantastic for us. Okay, so hopefully we'll get to there in time. But I just need to introduce you to a bit of rudimentary virology first. This is a flavivirus. It's an RNA virus, so its nucleic acid enters the cell and behaves essentially like a messenger RNA, encoding this complicated polyprotein, which is spatially organized into things that make up virus particles and into things that replicate the RNA genome and manipulate the cell and what have you. And the protein we're interested in here is the M. It's made as a precursor. It's part of the virion. Okay? So this is interesting because, as I mentioned, these channels act during both virus assembly and entry, so we wondered if that was going to be an important target. We do think there are other roles in the cell, but we're pretty convinced that this channel is going to be working during uncoating. Now, why do we think that? So this is, just a simplified virus life cycle. It replicates in cytosolic membranes, it manipulates the secretory pathway, and M actually has a very well-defined role as a chaperone for the envelope glycoprotein on the way out, and no one knows what it does on the way in, even though it's present in the virus particle. Viruses don't do things for no reason. So the fact that this is present in a virus particle suggests it has to do something. And we hypothesized that one possible reason for this could be during virus entry, as I mentioned before, in that kind of manipulation of the internal virion environment as the endosomes acidified during the progressive entry process. So this is a very highly ordered virus structure that flaviviruses have. And what you can hopefully see here is that the pink membrane proteins, the M protein, are associated with an E dimer, the virus glycoprotein. But what happens during endocytosis is this undergoes a transformation from a dimer to a trimer, and when you look at cryo-EM structures of these, you can no longer see the M protein associated with these envelope proteins. So when the fusioneric changes happen in the virus, this M protein is free to go about its business, we thought. So that was a good opportunity for it to rearrange, we thought, and make these oligomeric structures that could form an ion channel. So this is the M protein itself. It's very short and hydrophobic, as I mentioned, all these viroporins are. It's mainly got these two transmembrane domains, and we know this from the cryo-EM reconstructions of the virus particle, and this very long N-terminal domain here. And when we've done, just to orient you, when we've done in vitro work and molecular dynamics situation, we've cleaved off this disordered end of the protein just because we don't think it's relevant for the channel formation. And so what we wanted to do is just see, is this thing actually capable of forming a channel? And what we could find very quickly is that looking at M-encoded peptides, were forming in membrane mimetic environments. We could see that these were actually forming channel-like structures, which was kind of important. So these proteins on their own, left to their own devices, will form lots of different channel particles, and we wanted to do cryo-EM reconstructions on these, but unfortunately they're quite heterogeneous. So we couldn't do stoichiometry on them. But again, we could see by other methods, such as native page, that this protein likes to be an oligomer when it's in a membrane, unless it's being restrained by that glycoprotein interaction. So unfortunately, as I mentioned, we can't use electrophysiology for these proteins. So we use an assay that's an indirect dye-release assay, which I'm sure many of you would be absolutely horrified by. But actually, this indirect assay does recapitulate many of the properties that we see in electrophysiology for these viroporins as well. And what was nice is that, as well as in standard liposomes, this protein, this M protein, was forming pores that were able to cause dye release in standard liposomes, as well as sort of virion-shaped curvature liposomes as well. So certainly, this is a membrane-disruptive protein. It's also activated by reduced pH. And this is, again, if you think about its role during the endocytosis pathway, this could be important in terms of its activation and function. And thankfully, our big shotgun approach using romantidine showed that you could stop this channel forming. And we've got other data showing how wide these channels are from dye-release assays, et cetera, et cetera. So we're on to something, we thought. And so we hope that we actually have some relevance in trying to drug this protein during the life cycle. And sure enough, in cell culture assays, if you add increasing concentration of our dirty inhibitor romantidine, we can progressively and dose-dependently reduce the replication of these virus-infected cells, shown here in green, using an antibody against the glycoprotein. You can see that in a Western blot and down here as well. And what was really interesting and nice to see that sort of affirmed our hypothesis is that this drug works far better if you add it in the early stages of the virus life cycle. If you let the virus infection continue, then add the drug afterwards, it doesn't really seem to do very much. Okay, so that is consistent again with the idea that this is working during virus entry. But perhaps most importantly, we wanted to ask whether actually this would have any clinical relevance. And so we used a mouse model of Zika virus infection that we have available in Leeds with Clive McKimmey. And we transiently suppress virus interferon responses because Zika is a bit weedy in mice models. And we infect in the skin along with a mosquito bite to sort of recapitulate the natural infection with and without our drug and look at different samples later on. And what we found, again, was consistent with this idea that we're preventing the viruses getting in and out of cells. Whilst you can readily detect the virus at the injection site, which you might expect because you've injected a load of virus, when you actually look at the viremia, so the traffic of the virus around the body in these mice, even this dirty inhibitor, albeit added at a fairly high concentration, is absolutely pretty much preventing the anemia, so the spread of the virus around the body caused by the infection of these mice. So this is really nice, encouraging preclinical data to show that at least this virus target could have some relevance. So we wondered what these things might look like, and we used molecular dynamics simulation and protein docking techniques to try and get that cryo-EM-derived structure of a monomer to fit into an ion channel type structure. Now, of course, this is guessing, but actually we tried lots of different combinations and hit upon this hexameric channel structure with a sort of rotational symmetry forming kind of like a camera iris type formation. You can see here how an individual protomer fits in. The third alpha helix is fitted into the channel lumen, we believe, and you can see that there's a reasonably sized aqueous channel that forms through there. Not much in terms of constrictions, however, you do then see closure in simulations, which I'll show you in a minute. And I'm not going to talk about this in great detail, but we believe that these transmembrane spanning helices that line the channel lumen may have properties consistent with sort of hydrophobic gating in the ion channel complexes. And these are centered around a cluster of leucines in the channel complex here. So we wanted to see whether these might be stable, so we used MD simulations. And what was really pleasing to see, oh, no, both of them, stop. Oh, anyway, okay. Shouldn't have tried doing animation, should I? They didn't fly apart, which was the most encouraging thing. These things are stable, and they do eventually close around these leucines that I mentioned just before. And although it wasn't statistically significant, and we were only doing 200 nanosecond atomistic simulations, mimicking acidic pH by protonating histidines in the channel structure led to these things being more spontaneously closing more sooner, more early, if you like, during the simulation, so that you can't really make a judge of that, unfortunately. So we were kind of gathering evidence. We thought that these were viable targets and that we can actually try and drug this sort of channel activity, but where could we design rational inhibitors? Well, we used an add-on to the Maestro program to try and look at different potential sites for directing these inhibitors, and we found some, specifically this L1, so luminal region, and also on the membrane-exposed periphery of the channel, that look like good, viable, druggable targets. And we've also seen this in other viroporins. And these are actually basically located inside and outside of this cluster of leucines around where these channels are closing. So we used in silico HTS to derive a load of repurposed drug hits from various different libraries that we had available in Leeds. And this is just our sort of very simplistic pipeline that we use to develop new inhibitors. So we have our in silico HTS, and then we'd had a process of attrition and rationalization of these inhibitors that we identified. We then test them in vitro, and then in virus cell culture. And while we haven't got to this stage yet, because we need another grant, we hope to do iterative refinement to try and improve and understand the SAR of these sorts of inhibitors that we've identified. This is an example of our in vitro assay screens. The reason that we like using these dye-release assays is you can scale them up to 384 or 96-well formats very readily. And we've screened other viroporins against thousands of molecules in this way. But this is an example of our peripherally targeted screens. And what we could see was that we were enriching, even at just a 1 micromolar concentration, we were enriching candidates that had hits up to 20% through our rational process here. So that's far above what you'd expect at random. So we were really pleased with that. And I'm just going to tell you in the interest of time about our 2 so-called best hits, because obviously that's what you would do. And so what we were really pleased to see is that we could actually derive hits against both the luminal drug target and, as I'll show you in a second, the periphery. And these are repurposed hits, remember, but they're slightly strange. And we do believe that the effects that we're seeing against virus is specific because the canonical targets for these small molecules are absent from the cells that we're doing the virus culture in, okay? This is a docking simulation for these sorts of molecules that we're seeing hitting the lumen. And whilst the chemical formulae are very different from the hits that we had from this series, a lot of them are very similar in terms of these very long molecules that appear to actually attach to the lumen in more than one place, sometimes up to 3 places, in that pocket that's adjacent to those leucines binding. And similarly, in the periphery, we identified multiple different sort of planar molecules, which are similar to other molecules we found against different viroporins, which bind into this peripheral cavity I showed you before, again, adjacent to these leucine, we think, gating region here, and that sort of is putting a wedge in the camera iris and preventing it closing. And importantly, And we could see that this was dramatically improved compared to our original rimantadine screen, at least for the luminal targets, which we obviously have a bit more confidence about because of the hydrophobic penalties and things which we experience when trying to sort these in silico. So we're down in the sort of low nanomolar ranges for this repurposed HIT, and I emphasize it's a HIT. We don't have any other further chemical information about this. But as a last port of call, what we really wanted to do is just show that this is actually specific, that there's not kind of some kind of add-on or off-target effect in the cells. And so with viruses and these virion-resident channels, we can play a trick, which is to dose our virus particles and then separate them by ultracentrifugation. And you can plot the infectivity of the virus along these gradients when you fractionate them. And the nice thing is that it separates the virus particles and the infectivity from our unbound drug molecules. And when you do that, you can see that for wild-type virus, we see a nice peak of infectivity here. And for both rimantadine and our 2 repurposing hits, we see a significant decrease in the infectivity present within those purified virions. And when we normalize for the actual contents of those virus particles, you can see that the specific infectivity of those virus particles is being reduced. So we think that this is a sort of, at least if not irreversible, a certainly very favorable drug interaction that allows these viruses to be dosed and rendered non-infectious, at least for a sustained period over a cell culture assay. So I'll leave it there, and just to summarize, we believe that these viroporins are a good druggable target, even though we have this double-edged sword of the clinical precedent of rimantadine. We believe that this Zika virus M protein does form a virus ion channel during this rearrangement, we think, during virus entry. And we've identified 2 at least proof of principle sites against 2 different drug binding sites, which appear to be amenable to further SAR drug development. And at the moment, we're working with John and my other colleagues to try and get a handle on how these things are actually working. So importantly, I just wanted to finish by thanking Emma, who was the PhD student who had to learn virology and MD. She hated me, but it was brilliant. And then Richard Foster, who's our medicinal chemist colleague, and Andreas Kelly does all of the molecular dynamics, and John we're hoping to work with on the actual gating mechanisms of this. This is my group at a recent Viva celebration, and of course this was funded by the IAA, Funds for New Care I, and also the MRC. And I'll leave it there and take any questions. Thanks.
Questions? Damien.
That's lovely talk, Professor Griffin.
It's very formal.
I've been an advocate for exactly this idea of using small molecule or whatever type of molecules you can find to target ion channels, partly thanks to our discussions over the last couple of years on this thing. But playing devil's advocate, there's always a but, and maybe even thinking about government pushback, because they're clearly the ones that we think should be funding this kind of research.
Yeah.
Since the mRNA vaccines were probably beyond most virologists or vaccine experts, you know, success capabilities in terms of these mRNA vaccines that were created for SARS-CoV-2. Mm-hmm. Where do you think this kind of drug discovery would fit into that paraphernalia as an antiviral?
Well, I think the best use of antivirals, you know, if you're talking about an emerging pandemic scenario, it's very early on. So, you know, for example, antivirals specifically, if you also have the possibility of prophylaxis, it's the early stages and treating those patients that are the most severely unwell. So, you know, if you think about COVID, the first year of COVID, however many millions of people died, even with mRNA vaccines, it still takes time to roll those out, to get the right target. And, you know, there's no guarantee that they work. So I think we all sort of take for granted our COVID vaccines now. There had never been an effective coronavirus vaccine, whether in humans or in animals, prior to those vaccines. So we were lucky, okay? Flu is probably a different question. We know we can make flu vaccines. But still, as swine flu showed us, that is quite capable of spreading across the planet prior to any vaccines being rolled out. So I would argue that this antiviral prophylaxis idea is to treat those most vulnerable patients, which, by the way, don't often respond to vaccines, and as well to limit spread in the initial stages of an outbreak.
Thank you.
Hello.
Any quick questions?
Yeah, I can. Hello. Hi.
Okay.
Yes, no.
Hi. Excellent talk. I've got really quick 2 questions. One is...
One
Okay, one question. That's fine.
One quick.
But I can find
Pick
it
the
later on.
hardest.
The remanded, I'm not surprised that you have to use a high concentration given the size of the molecule.
Yeah.
But do you think at that higher concentration on its own, can it also change the pH of the media, for example?
Oh, so we controlled for that, yeah. So we used buffered media and we looked at andesitic uptake and it wasn't affected. So yeah, that's
Thank you.
just didn't show that in time. Yeah.
Great.
Okay.
Thank you so
Thank
much.
you.